All right, we're going to get started. So welcome everybody to this first innovation showcase of our winter series entitled Charting a Course for Clean Water, Mitigating Organic Chemical Contamination in the 21st Century. My name is Robert Nyman. I'm the Manager of Partnerships and Programs at the Ontario Water Consortium. The Ontario Water Consortium is Ontario's only sector support organization dedicated solely to water innovation. We bring together researchers, companies, municipalities, and governments to drive collaborative R&D, pilot testing, and real-world technology adoption. We help bridge the gap between research and market-ready innovation. And while there are many innovation organizations across Canada, none are focused specifically and exclusively on water like OWC is. OWC's collaborative model delivers measurable results. Over the past decade, we've helped mobilize over $90 million in project funding, supported or created more than 350 industry jobs, and advanced nearly 80 water technology projects. We've partnered with more than 100 academic researchers and helped build infrastructures, including wastewater test beds in London and Guelph. We do this by connecting innovators with adopters and aligning the right funding, partnerships, and support to move solutions forward. Before introducing today's webinar, we also hope you'll join us again for the rest of our winter webinar series. On February 17th, we will hear from the University of Guelph's Morwick G360 Groundwater Research Institute, showcasing new research and practical tools for characterizing and monitoring fractured bedrock aquifers to support source water protection and sustainable subsurface energy development. Then on February 26th, Toronto Metropolitan University will present plant-wide modeling approaches for wastewater treatment plants, highlighting diagnostic tools for managing fugitive methane emissions. I'll drop the registration link in the chat in just a moment. All sessions are free to join, and please also note the QR code that you can use to subscribe to the OWC newsletter. Now, I'm very pleased to introduce Dr. Maricor Arlos, Assistant Professor in Civil and Environmental Engineering at the University of Waterloo, to begin today's webinar. Dr. Arlos will provide an overview of integrated approaches her research group is advancing to understand and address trace organic contaminants in water system. Following Dr. Arlos, we will hear from Jingya Pang, who will discuss innovative strategies for repurposing industrial byproducts to remove trace organics. Melissa Bennett, who will present an integrated modeling framework for pesticides, fate, transport, and bioaccumulation. And finally, Mackenzie Rosebrugh, who will share new insights from bioanalytical assessments for stormwater impacts on source water. Maricor, the floor is yours.
Thank you very much, Robert. Let me just share my screen here. Can folks see my screen?
Excellent.
Okay.
We can see it.
Perfect. Thank you so much for inviting us to present to the OWC Innovation Showcase Winter Series. I'm very excited to present the work that we have been doing. I just recently moved to University of Waterloo starting fall 2024, so really happy to share some of the preliminary work that we are building here at the university. So before I begin, I do want to first do a land acknowledgement. Much of our work here at University of Waterloo takes place on the traditional territory. Is it not moving? Okay. Let's see here. I
can try and let me see if I can move it. I was able to move it on my end. Are you able to take control?
I thought I have been taking control, and it looks like, yes, okay, now I can see it. There you go. Thank you.
Yeah.
Um, right, so I just wanted to acknowledge again that much of our work takes place on the traditional territory of the Neutral and Anishinaabe and Haudenosaunee peoples. Our main campus is situated on the Haldimand Tract, the land granted to the Six Nations that includes 6 miles on each side of the Grand River. So before I begin, I just wanted to sort of give everyone an overview of the work that we're doing. Interdisciplinarity is really a main theme in our work. We believe that water transects multiple disciplines, meaning that we need multiple different perspectives to answer water solutions. So the area that I've been trained on and the things that I've been working on maybe for like since I started my master's and also for the last 10 years is exposure science and environmental risk. So this is looking at the fate and transport of substances in the surface waters and their impact on aquatic organisms, mostly looking and delving into environmental science and aquatic ecotoxicology. The second part, and maybe I would say 30% of the work that I'm doing is on sustainable treatment. So right now we're partnering with many partners to develop nature-based solutions, also using waste materials for water treatment. And finally, the area that I'm slowly starting to incorporate in potentially my career is knowledge co-production. I am continuing to develop relationships with Indigenous and equity deserving groups to co-create and develop solutions for water security. So these are the main themes that my research group what's basically identifies ourselves as focusing on any of these themes. Primarily, we look at the urban water cycle. So for many people, we live in an urban city, and it mostly begins with the source water being extracted from a river, groundwater, or lake systems. And then we go and then treat that, distribute that to the entire community. We use that water multiple different ways, domestic, agriculture, or industrial. And then it goes into wastewater collection, then again, back to the source water, probably likely multiple kilometers downstream of where the source water intake is. And within that also there is an inputs from stormwater and also some industrial discharges that are not necessarily, maybe not connected to the system. But during this process, when the water moves around, it also carries with it some chemicals. And that's where the focus of our work is on, primarily on micropollutants. So micropollutants, we define them as substances that are present in the environment at extremely low concentrations. When we say low concentrations, we talk about parts per trillion, nanograms per liter, or a drop in a lake type of concentration. So that includes a variety of different types of chemicals, including pharmaceuticals, personal care products, natural and synthetic hormones, pesticides, and industrial chemicals. And right now, I think the hot topic, micropollutant, is our PFAS substances. And it slowly, or not slowly, but it's always sort of being incorporated in our daily lives, like news related to medications leaking into waterways. And this is an example of a news article that talks about the impact of antipsychotic drugs impacting the migration of fish, primarily salmon. And if you look in multiple different places, you see, contaminants of emerging concern, sunscreens, microplastics. You can see, fentanyl and drugs of abuse in our waters and our wastewater systems. So they are being I guess this is not something that is just a hype. It does impact our daily lives and especially water qualities, given that there's going to be some impact of climate change and continuously deteriorating water quality in source waters as well. So for us in our lab, when we look at the presence of these substances in the environment, we look at 3 different tools. So the first one, which is the gold standard of exposure assessment, is through analytical instrumentation. One of the reasons that I moved here to Waterloo is access to these really fantastic instrument called the LCMSMS, and I just recently got mine. It's running 6 months, and my students are really happily running it. So this is the gold standard and able to basically measure substances at extremely low concentrations. The bioanalytical tools, which are mostly focused on the potential of that water to create an impact or create an effect. So we don't necessarily look for a chemical X, Y, Z, but we look for a chemical, the cumulative effects of stormwater or wastewater, for example. The third area that we use, or third tool that we use, is through modeling. And modeling can come in a variety of ways, but typically we use the data from our analytical instrumentations, the bioassays, to inform our models, to predict concentrations in the future, to predict different types of conditions in the future. And it can come in many different ways in terms of complexity and also simplicity and the use of potential users. So I'm really excited. Some of my students are here to talk about the different areas of research that involve these different types of tools. When we look at water quality management, one thing to consider is that site-specific conditions do vary. So for instance, I started out in Grand River Watershed. I was still a baby grad student at that point, and I moved to Switzerland, learned about the way the other people look at and value their resources. When I was at University of Alberta, Athabasca River and Bow River were sort of like my backyard. That's where I go and do my analysis and work. And now I'm really happy to go back to the Grand River Watershed again. I'm reconnecting to my roots and reconnecting to the region of Waterloo. And one of the first things I did was to contact Ontario Water Consortium, actually, to kind of initiate that network again, and revitalizing my relationship with the Grand River Watershed. So one of the things that really was a very important thing for me when I was starting out was when the fish, my former supervisor, Mark Servos, collected some fish downstream of a upstream of a wastewater treatment plant. So this is a rainbow darter, really healthy fish with 2 lobes of male testes, really healthy male here, and then found fish that is intersex downstream of a wastewater treatment plant. So if you notice these yellow blobs here, these are actually eggs or intersex fish that is associated with the wastewater treatment plant discharge. So at that time, it was a lot of the data suggests that the presence of estrogens, ethinyl estradiol, or birth control pill ingredient may be responsible for the intersex that has been observed. Over time, in the last 10 years, there's so many upgrades and so many changes in the region of Waterloo, and very happy to see and hear from Dr. Mark Servos that the intersex now is almost back to the baseline levels. That means the upgrades that they've done at the wastewater treatment plant not only removes nutrients, but also removes a number of things, including emerging contaminants, trace contaminants, or micropollutants. So that's sort of where it started for me. And when I look at my research program and the work that we do here, one challenge is figuring out where these chemicals are. I often tell my students that we are sort of like environmental forensics. So we figure out where the chemicals go, what they do when they get there. And the second piece that I also wanted to incorporate and let them know is that there probably is a way for us to reduce our exposure and also the risk of exposure to these types of substances. So that brings me to a few emerging regulations, and one here is the European Union. They're probably very progressive in a way that they have pressures of population growth. They have large or high urban densities, and it makes sense for them to do certain regulations that may or may not apply to the North American context. But I'll give you my thought and opinion about that in a little bit. But first, the European Union basically says, hey, all wastewater treatment plants that service 150,000 people, you need to treat your wastewater, and you want to make sure that these indicator compounds, so if you look at all of these 12 substances, amisulpride, citalopram, diclofenac, candesartan, metoprolol, venlafaxine, these substances need to be removed at least 80%. So they have mandated this in their Urban Water Framework Directive, and they wanted, the goal is by 2045, all of those treatment plants have now upgraded it to remove these substances. In terms of technology, though, they are focused on ozonation or activated carbon. So these technologies are not necessarily applied in Canadian context for wastewater treatment. They require a lot of energy and they also require a lot of cost. So something to think about from a Canadian context where people, so you see ozonation and activated carbon in Canada in drinking water treatment plant, but not necessarily wastewater. So something to think about here also in terms of, technologies for removing these substances. We recently wrote an open access government article with my mentors, Dr. Kelly Monkettrick, Mark Servos, and Fred Rona from University of Calgary, and Marcus at Waterloo. And we basically look into develop an opinion related to how can Canada sort of see or change the way we see water. So we have, everyone says it's solution to pollution is dilution, but eventually we'll need to realize that our water resources in Canada are not unlimited. So we see lots of data showing that our water quality is deteriorating. So we really need to do something about improving our water systems, not just through technology, but also maybe in the upstream side, like the source management, for example. So I invite everyone, if you are interested, it's just really a 2-pager, quick, it's mostly written for regulators and people in the government to look into what might be good decision making moving forward when it comes to pharmaceuticals and personal care products. In terms of treatment, I am very happy. We just got a funding from Alberta Innovates to look at nature-based treatment to remove or to improve dairy wastewater. So right now that's ongoing. It's a 5-year project, and I have a master's student starting in May. Very excited, but really again looking into what we can do to water reuse and removing different types of not just nutrients but also substances of emerging concern. And the most recent one, and I'd like to share this, and Jingya, my student, will also present, is the use of bauxite residue as a potential adsorbent material for wastewater. So bauxite residue is a waste material, and when the company or industry partner reached out to me, I sort of did not believe that it is possible to use this as an absorbent, but when we did a lot of bench scale testing, I really can see some potential here. And now I want to hand it over to my PhD student, Jingya, who will now talk about bauxite residue and her project related to this substance.
Okay. Hello, everyone. Thank you very much. My name is Jingya, and I'm a PhD student now. So my research project is focusing on repurposing bauxite residue as an adsorbent for removing trace organic contaminants from water. So let's start with bauxite residue. It's also known as red mud, and it's a byproduct from the Bayer process. After alumina has been extracted, the residual content is called bauxite residue. To produce 1 ton of alumina, about 1 to 2.5 tons of bauxite residue will be produced, and with now a global generation of approximately 150 million tons per year. Bauxite residue primarily consists of oxides, including ferric, alumina, silica, titanium, sodium, and calcium. As shown in the image, the red color of bauxite residue was given by its ferric oxide composition. And in recent years, red mud has attracted growing attention, not only because of the huge volumes, but also because there is an opportunity to recover metals and create value from this waste. At the same time, in Canada now, there is a broader push to rely less on external supply chains and strengthen domestic productions of critical materials. If alumina and related production expand, then bauxite residue generation will also increase. The current method of managing bauxite residue is storing it in the tailing ponds as shown in this image too, and this practice is exactly what drives many environmental concerns. First, bauxite residue is produced and stored in huge amounts, and it is highly alkaline, pH ranging from 10 to 13, so this results in the potential to pose hazardous and leach out toxic chemicals into the surrounding environment. Note that use bauxite residue as an adsorbent and activating process is always necessary to improve absorption capacity and reduce potential toxicity leaching. So in our work, raw bauxite residue undergoing a reduction roasting process, which is patented by our industrial partner GROWN, was turned into ABR. The color also goes from the original red to black. We also work with ABR in two physical forms: a powdered form for current bench scale experiment, and also a pellet form that is more suitable for pellet scale configurations. ABR is the baseline material for our study, and this is where our research started. Now, let me briefly summarize what we set out to do. Overall, our goal is to develop and translate modified activated bauxite residue, also called ABR, into a practical absorption approach for removing trace organic contaminants from water. We focus on three objectives. First, we develop an acid-assisted microwave modification method to tune ABR and enhance its absorption capacity. Second, once the optimal modification condition is identified, we will optimize absorption conditions, not only based on removal performance, but also by considering water quality impacts, so the approach is realistic for treatment applications. And then third, we assess the feasibility of integrating ABR absorption into existing wastewater treatment infrastructure, thinking about how it could actually be implemented. This image shows the workflow of my research. First, we aim to tailor ABR pore structure and surface chemistry through microwave heating plus acid treatment. Because microwave heating is an emerging approach for material modification, it is rapid, energy efficient, and can increase surface area and porosity significantly. Then, ABR surface property will be assessed using multiple characterization technologies, such as VET analysis for specific surface area, ICM for morphology and pore structure, and zeta potential for surface charge, etc. Next step, the absorption efficiency of ABR using a mixture of PFAS will be tested. ABR will be evaluated for removal efficiency, cell toxicities, alkalinity introduction, and the optimal operation conditions will be defined. More co-occurring trace organic substances will be introduced too in this step. After confirming those requirements and optimal reaction conditions, experiments will be advanced to pilot scale testing to evaluate the compatibility with existing wastewater treatments. This result will be provide estimate of ABR settling behavior and also the solid production, further guiding sludge separation and operational needs. From our previous study, we have already seen some exciting results. This bar chart summarized PFAS removal by ABR and the different dosages and different contact times. At a relatively low dosage of 10 grams per liter, we achieve 100% removal for long-chain PFAS, and the absorption reached equilibrium within 1 hour. However, 10 grams per liter is still considered high for practical wastewater and water treatment. So to be more competitive and sustainable, the dose still needs to be reduced. This highlights a key tension from a water treatment perspective. We want absorption technologies to work at low dosages to be practical, but at the same time, we also want to create useful reuse pathways for the growing supplies of bauxite residue. And also in terms of the modification, with hydrochloric acid assisted microwave modification, the surface area of ABR increased by up to about 18%, and we also observed shifts in surface charge, which can affect interactions with charged organics. So this can lead to the preliminary absorption test using 3 organic deaths as representative module trained organics. These results suggest that the performance of ABR is not only driven by surface area. Electrostatic and functional groups interactions also play important roles. These insights also guide our next step, tuning ABR to better target charged trace organics and compounds with specific functional groups. Now, I'll wrap up with our next step. I'm still working on optimizing ABR modification conditions to constantly achieve the surface properties we want. And then the removal performance of modified ABR will be tested using our new LC-MS instrument. We have already established robust methods for 21 pharmaceuticals and 24 PFAS. After that, we will toward more realistic water matrices and include competitive absorption effects. Finally, we will evaluate pilot-relevant configurations, including how to implement ABR absorption and how to handle solid-liquid separations. This will help translate the material improvements into a practical treatment approach. So thank you, everyone. And my talk looked at how we can remove trace organics using an engineered adsorbent. And next, Melissa will take us upstream with a modeling perspective.
Awesome. Thanks so much, Jingya. Yeah, so I'm going to be looking at movement of pesticides through the ecosystem. So for a little bit of background, hold on. I have to remember which steps I'm doing. There we go. I forgot that I had to take control of the presentation. Technical difficulties along every step of the way. It keeps it interesting. So modern agriculture is really large and relies on chemicals like pesticides and fertilizers to help keep up their outputs. Pesticide use does have hazards, some that we know about beforehand, like that we learn in production, and some that we don't know about. Modern pesticides aren't supposed to bioaccumulate, meaning that they don't stay in organisms and they shouldn't move their way up the food chain, but that doesn't always happen. So 2 studies, including one by our partners at AOAG, which is the Swiss Institute for Aquatic Science and Technology. They're in Switzerland. They're very cool. They've looked at internal concentrations of pollutants in gammarids, and they found that they were much higher than anticipated by the water concentrations. So gammarids are really small aquatic invertebrates. They look like a little shrimp. So just think of like freshwater shrimp, and that's more or less what you've got. These little guys are sensitive to aquatic pollution, so they're often used as subjects for water quality. So the diagram that I have on the right side of the screen, we start here with like a tractor spraying fertilizer onto crop, and if we follow the lower arrow, after a rainstorm or something similar, moves pesticide from that crop into the waterways, or it can blow through the air onto the trees nearby or onto the roots where it picks that up, grows into the leaves themselves, which also drop into nearby streams. Those things can both flow downstream into here where we have our little shrimp gammarid, and those things that can take into the pesticides into it through the water, through sediment, through the leaves, and it can also the pesticides can move into the sediment, and the gammarid can also get rid of some of the toxins itself. So what is the objective of the study I'm working on? Our study objective is to determine which exposure pathway is the main driver of bioaccumulation in the gammarids, figure out why those internal concentrations are so high. We're looking at water, sediment, biota, or possibly another source or pathway we aren't sure this time, which is what my little diagram is saying, pointing to those different methods, and then this little question mark in case there is something we haven't predicted yet. Our current hypothesis is that hydrology and the application rates of pesticides are the major drivers of this bioaccumulation. We also want to look at sediment and organic matter, so those leaves, because we believe they are important in intake pathways. for this. So why is this research important? Most of our water quality guidelines are and regulations are based on concentration in the water. But if water concentration isn't actually predicting impacts on our organisms, is it really an effective regulatory tool anymore? Or do we need a multi-pronged approach to protecting the environment? It's important questions we need to ask. It also shows us that we need to continue to study compounds, things like pesticides or fertilizers that have been developed and put into production recently. One of the pesticides that we're looking at, fluoperam, was only approved in use in Canada in 2013. It's only been about a decade. It hasn't been around as long as some other compounds that we might know more about. So, getting to the design of the study itself. We have 3 sites in Switzerland, which are these little yellow dots in the top. The Analog Lab has gathered the samples from these sites and then analyzed it for their pollutants of interest. So all 3 of our sites are in northern Switzerland. There's lots of agriculture in there. The Alps are in the south, so if you're thinking about those, unfortunately, that's not where my study is located. But there are lots of little streams all across Switzerland. They feed into a few larger ones, but we're looking at the little guys from our study. The smallest is Schellesbach, which is this guy here on the far right. It's only about 2 kilometers squared. It's quite small, but there is a lot of things to study in it. It drains into Lake Constance. I believe it's on the border of Germany. Serb is our next largest site, which is here at the bottom, around 30 kilometers squared. And then our largest is Halbach in this like most northern region of Switzerland, and it's around 75 kilometers squared. So the first part of the research plan involves building a hydrologic model. What this does is allows us to predict hydrologic conditions, so how much is going to be in the river from rain, from no rain, that kind of thing. Allows us to predict how much water is in the system, allows us to predict future conditions, and we can also add in climate change scenarios if that's something we want to do in the future. The image here on the screen is of the computed watershed of Schellesbach, so the really tiny one. I made it using land use data, some elevation data, and then the computer uses that to figure out where the water will flow to, which is very cool. After building this, the model needs to be calibrated and validated so it gives out good results instead of just random numbers. So I'm currently working on the calibration step, and then we'll move on to the next step of the project, which is pesticide loading rates. We want to know how much pesticides are being applied on the fields nearby these streams and what types. This will help us understand how much contaminant is entering our receiving environment, that is our streams, and what type of pesticides, and confirm if the pesticides match our sample results. Just make sure that we're understanding the same things. After this, we move into building an in-stream kinetics model. So this helps us understand how contaminants move in the environment and where they go when they enter the streams. Using kinetics data and properties of the compounds, we can understand if the compounds stay in liquid form, dissolve in the water, partition sediments, and how those things balance out into equilibrium. And then lastly, we'll build a toxicokinetic model to understand the bioaccumulation. This does something similar to the in-stream kinetics model, but it's more for organisms. We can take it to model how much the ammonites will take in from each source and how much they will remove or depurate from their system. From here, we can determine if our hypothesis matches the mechanisms in play and if these processes explain the bioaccumulation, which is the overall goal of this. Last thing I'd like to leave you with is that this is not the typical approach for this kind of project. Often research is segmented between the different stages, so hydraulic modeling, in-stream kinetics, or toxic, like a bioaccumulation model. It'll sometimes include one of these or 2, but not usually all of them. So this project is very interdisciplinary, starts at the source and ends at the expulsion of waste from our affected organisms. It's a little more comprehensive than your typical project. It's also worth mentioning that while the study its sites in Switzerland, it's still really applicable to Canada. We both really value agricultural output, and we both use pesticides on our crop to maintain that output. So we can still learn a lot of information that we interpret from this study. So thank you all for listening, and I will pass you off next to Mackenzie.
Thanks so much.
Yeah, especially if we're gonna stop.
So I will be focusing more on the impacts of stormwater on a source water reservoir. And for this project, we partnered with the city of Calgary. So Calgary water system consists of 2 drinking water reservoirs. First, there's the Bearspaw Reservoir, which is fed by the Bow River, and the Glenmore Reservoir down here, which is where my project is located, and it's fed by the Elbow River. Calgary has a mixed-use watershed, so people use it for recreation as well as source water.
I just went through our email template.
The Glenmore Reservoir is located on the Elbow River in the southwest part of Calgary, supplying about 40% of the city's water needs, but there hasn't been a lot of change to point sources with the changing urban landscape. Partnering with the city of Calgary, the city monitors 5 catchment outfalls draining into the Glenmore Reservoir. These outfalls are located here. The red dots are the current monitor rates, and this one has some data, but it was shut down in 2018. The data collection started in 2017, with their main driver being source water protection. The research I'm doing focuses on how the stormwater affects the loading of contaminants in this reservoir. There is research being done on microplastics and heavy metals and other contaminants, but I'm working on a model development for loading of like more basic contaminants and then measuring the toxicity.
And it is mostly a hyperlink from us in this link, so it's not.
So the first
so if it's already
kind of portion of our study is the loading calculation. Using the stormwater management model that's currently being developed, I'll determine the pollution contribution of stormwater in the Glenmore Reservoir. Next, we'll evaluate the toxicity by using bioanalytical methods to measure toxicity of stormwater collected since 2024 and their relationships with hydrological events. And then finally, we'll assess the relative contribution of 5 subcatchments on the water quality of Glenmore Reservoir using information from the model and toxicity results.
So that is something that we like, obviously, I mean, not.
So our first objective was to design a model to estimate the flow rate and contaminant load of each outfall. This was done by entering online land use data, topography, catchment areas, and pipe systems into ArcGIS, which gave us a base model that can then be put into SWIM along with historical rainfall data to simulate flow during rain events.
That's it.
Once this model is calibrated, which I am currently working on, a loading will be estimated based on the simulated flow and the concentration data.
Yeah.
The parameters in the model will be representative ones that are present during base flow and rain events.
Yes.
Currently, we have data from the past years showing relationships between the concentrations of contaminants and rain events. The data we were given was a set of conventional parameters, so TSS or chloride. This model shown on the right has been established in SWIM, and I am currently working on calibrating it with the flow data that was provided from the city of Calgary throughout their water management program. So the data we have from 2017 to 2022 that's being used to develop the model is just the tip of the iceberg. These are the special parameters with known effects. However, there's a lot of unknown chemical risks, which is where we can start looking at toxicity. The bioanalytical methods look at cell toxicity pathways to determine effects of the stormwater samples. Yeah, sorry, I have some people sitting around me also in meetings.
Graham really should have thought about it before he made something too.
So there are 3 different types of toxicity tests: non-specific, relative, and specific. A general rule when conducting these assays is to have a representative of each one. So for our toxicity analysis,
we chose a cytotoxicity, which determines toxicity based on cell survival. The reactive will be mutagenicity,
that's what I would like.
which determines toxicity based on cell mutations. And then for specific, we will do estrogenicity to measure the estrogen levels of each sample. So going on to our next steps, I'll continue working on model refinement to validate the model using flow data collected at each monitoring point and add the contaminant data to the model for load calculations. Then starting in May, I'll be running bioassays on the samples from the current and past years to determine toxicity. And sometime around the next year, I should be able to run the model with the data from the bioassays and determine any links between flow or contaminant loading with toxicity of the reservoir. And with that, I'll hand it back to Maricor for closing. Thank you.
Thank you very much. And thanks to everyone for your patience with a lot of the things that are happening here with audio and also some technical difficulties. But I do want to thank the people that have been very, very helpful in building my research program, especially the students, those from University of Alberta and now the students at University of Waterloo. Very thankful to a lot of our funders and the institutions that helped me shape who I am today. And with that, I'd like to end our webinar and let's make waves together. And we're happy to answer any questions that you have. Thanks again for your patience.
Thank you, Maricor and team and everybody. Great presentation. We now come to our Q&A section of our webinar. So you're welcome to put some questions in the chat, or you could also alternatively raise your hand and I can unmute you if you want to ask the question directly to Maricor or any other students. So that's open now for you. There's also the Q&A function if you want to use that or prefer to use that, either way. So we have a question here coming from Mark Payne, and it's for Melissa. And the question is your bioaccumulation model going to be fugacity, I don't think I'm pronouncing it right, based?
Fugacity, yeah.
Fugacity.
Yeah.
My apologies.
I'm sorry, I still think I missed that last word.
Fugacity based. Is your bioaccumulation model going to be fugacity based?
I'm gonna hand that one over to Maricor. Since I'm still pretty new to this, we haven't other than kind of drafting out the main steps, we haven't gone into major detail on what those steps entail. And I am honestly not super familiar with that word. So I'll maybe pass it over to Maricor because she might be able to answer that better.
Sure. I'll take that over. Thanks, Mark, for your question. So Melissa right now is only in her 6 months of her master's. So she's really, really just fresh and out. So right now she's in the hydrology section. For the bioaccumulation, we foresee it as something more toxicokinetic. So uptake rates, depuration rates, and also some rates associated with exposure to sediments and maybe dietary intake. So we have a different model for a bioaccumulation / toxicokinetics model for Athabasca River that we are developing right now, which hopefully we can apply to our sites in Switzerland, and hopefully they're transferable. So I hope that answer some of your questions there.
Great, thank you. Thank you, Maricor. There was also a request to end your share screen just so we can maybe see everybody's faces as they respond a little bit more.
Okay,
clearly.
yes. So I would just stop sharing. There you go.
Yeah. That's great. Perfect.
Thank you.
Then the next question that came in is from Marius Bauer. Could bauxite residual be used as PAC/GAC? Being used?
Sorry. Yeah, so that's another problem we are working on. So in my master's work, I have already compared the efficiency of ABR with commercial available powdered activated carbon. So the reason why we chose the 10 gram per liter as a baseline, because the 10 gram per liter is already comparable to 0.1 gram per liter of PAC. So unfortunately, the dosage is still very high. So that's why we want to further modify ABR surface area and increase its absorption capacity. Then we can reduce the dosage and really apply ABR into our current existing wastewater treatment technologies.
So I just wanted to add, thanks, Jingya. So bauxite residue is a waste material and they have tons of it. And we are going back and forth with our industry partners because they think, oh, it doesn't matter if we need to use high dosage because we need to get rid of them. But the other part is like, well, there's a lot of issues with how do you separate that later on in the waste stream. So the lower the dosage is better. So PAC and GAC right now are still considered the top in terms of absorption. And we, so part of Jingya's project is actually, like she said, modifying it more to improve the absorption capacity. It may not reach exactly as what PAC and GAC do, but they it's an opportunity to get rid of that waste. So for really high industrial waste and something, because once you get rid of that waste, you can actually recover minerals from there. We can deal with anything that's stored on there, and then it can be used further as a material for roads, aggregate material for construction within the industry. So that's the goal later on, is not really have any waste after the PAC or after the ABR has been used.
Great, thank you, Maricor. There's another question from Mark Payne, and this one is for you and Mackenzie. Which bioassays specifically are you thinking of using? What bioassays do you recommend for assessing public health risk and drinking water?
I can take this, Mackenzie. I know you're at the airport, and you can add more if I missed anything. So for the bioassays, the idea is you need to have a representative for each of the toxicity pathways that you're looking at. So if you're looking for non-specific, so any cytotoxicity assays will do, both mammalian cells or bacterial cell lines. And if you want to look for reactive, that's when, you know, a lot of assays like UMU assays, or so I'm not thinking top of my head here. It's the very classic carcinogenic assay, carcinogenicity assay. That's very reactive piece. And then the other piece, which is specific depending on the endpoint that you're looking for. So for drinking water, typically you would want to have a representative for each of those pathways. So specific, non-specific, and reactive. So drinking water, depending on the use, the end use of it from a public health perspective, the bioassays can actually be used. The genotoxicity assays, that is the UMU assay, it's available right now commercially too. You don't have to create that in your lab. It can actually detect the potential of your samples to cause carcinogenicity. So that's for drinking water from public health perspective. The impact here is mostly for if you have any issues with disinfection by products, if you have any treatment plants that have high organic matter source water, that will then impact the production of disinfection by products. So I think that's where that would be useful, but typically you would want to have 3 of each representative for whenever you're doing water quality assessment using bioassays.
Great, thank you, Maricor. And here's one for Melissa. And it says, if I understood correctly, you said most of the work in this area approaches 1 or maybe 2 of the parts of the system, but usually not all 3, while you're taking a more holistic view. Does prior work look at just one part of the system just because it's easier, or are there any limitations that come with looking at the whole system?
From my understanding, some of the limitations come from either not looking too deeply at one aspect of it or not having the expertise to look at the whole system, because we have both engineers who understand like the hydrologic pathways, and we have the folks at AOWOG who are really good at the toxicokinetic side, like the bioaccumulation side. We can really integrate both of these pathways together and look at that chain together so that we don't have to do two separate studies and we can combine it into one. There are probably other reasons beyond just not having the expertise, but that's my current understanding of it. Maricor, if you have any other thoughts, please feel free to chime in, but that's kind of what I've got for now.
That was great, Melissa. Thanks for responding to that. Yeah, so a lot of our training is usually siloed. So engineers are not necessarily trained in a toxicokinetic bioaccumulation part of the model. Maybe the fugacity part model comes in at a later stage, but we are really trained on the hydrology and then the in-stream fate and transport. And the mechanisms for bioaccumulation, a lot of it is being provided by toxicologists and exposure scientists. And I think there's that opportunity for us to kind of meld those ideas together and probably create a framework that coherently goes from the source all the way to the receptors, like what Melissa said at her presentation. But yeah, I don't have anything more to add. I think you covered it well, Melissa.
Thank you, Melissa and Maricor. Are there any further questions from anybody? Again, you can place them in the chat, or you could raise your hand if you like. We'll wait a few minutes for some more questions to come in. We have about 5 minutes left in the webinar for any final questions. Well, if there's no further questions, then I want to thank you, Maricor, and the rest of your team, Melissa, Jingya, Mackenzie, everybody for presenting today. And it was a great webinar, very informative. And wishing you all a great rest of your afternoon. If you want to connect with Maricor and her team, of course, you can do so and talk further. And we can help you connect as well. If you want to reach out to the Ontario Water Consortium, we can connect you with Maricor's team as well. So thank you very much, everybody, and I wish you a great rest of the afternoon.